Mendelian inheritance, also known as Mendelian genetics or Mendelism, is a scientific description of how hereditary characteristics are passed from parent organisms to their offspring. It underlies much of genetics. This theoretical framework was initially derived from the work of Gregor Johann Mendel, published in 1865 and 1866. It is summarized into three laws: the law of dominance, law of segregation, and the law of independent assortment. The law of segregation states that every individual possesses a pair of alleles for a particular trait and that each parent passes a randomly selected copy of only one of these to its offspring. Whichever of the two alleles in the offspring is dominant determines how the offspring expresses that trait. During the formation of gametes or sex cells, these two factors separate. Only one ends up in each sex cell. On the other hand, law of independent assortment describes how different genes independently separate from one another when reproductive cells develop. Independent assortment of genes occurs during meiosis in eukaryotes.
Inheritance. The speculation that chromosomes might be the key to understanding heredity led several scientists to examine Mendel's publications and reevaluate his model in terms of the behavior of chromosomes during mitosis and meiosis. In 1902, Theodore Boveri observed that proper embryonic development of sea urchins does not occur unless chromosomes are present. That same year, Walter Sutton observed the separation of chromosomes into daughter cells during meiosis. Together, these observations led to the development of the chromosomal theory of inheritance, which identified chromosomes as the genetic material responsible for Mendelian inheritance. Figure A is Mr. Walter Sutton, and Figure B is Theodore Boveri. They are both credited with developing the chromosomal theory of inheritance, which states that chromosomes carry the unit of heredity. The chromosomal theory of inheritance was consistent with Mendel's law and was supported by the following observations. During meiosis, homologous chromosome pairs migrate as discrete structures that are independent of other chromosome pairs. The sorting of chromosomes from each homologous pair into pregametes appears to be random. Each parent synthesizes gametes that contain only half of their chromosomal complement. Even though male and female gametes differ in size and morphology, they have the same number of chromosomes, suggesting equal genetic contributions from each parent. The gametic chromosomes combine during fertilization to produce offspring with the same chromosome number as their parents.